Welcome to this video on metagenomic data analysis, where we explore the hidden potential of microbial communities. Metagenomics is the study of genetic material recovered directly from environmental samples, bypassing the need for cultivation. This approach is culture-independent, comprehensive, and enables systems biology studies of community-wide interactions. Our core research objectives in metagenomics are organized into 4 areas: biodiversity, ecology, function, and discovery. These objectives focus on quantifying diversity, mapping interactions, decoding capabilities, and identifying novel gene sequences. We begin with the first objective: community diversity. Taxonomic profiling addresses the question of who is there in a microbial sample. By aligning genetic reads against curated databases, we can accurately determine the abundance of various taxa. We analyze 2 main biodiversity patterns. Alpha diversity examines within sample richness and evenness. Beta diversity measures the between-sample differences in species composition, which is crucial for ecological community analysis. The second objective focuses on ecological relationships. Metagenomics helps construct co-occurrence networks. By correlating taxa abundance, we infer potential symbiotic, competitive, or predatory relationships within the microbial community. Niche differentiation is also investigated, looking at how environmental parameters such as pH, temperature, and nutrient availability influence community structure and specialization. High biodiversity is linked to ecosystem stability and resilience. This enables the community to recover faster from various environmental disturbances. Next, we explore the third objective: metabolic potential. Functional gene annotation uses databases like KEGG, SEED, or Metaseq to map genes to biochemical pathways. Metabolic potential is calculated as the sum of genes and pathway coverage. Pathway reconstruction identifies key components to infer the community's roles, such as carbon cycling, nitrogen fixation, and bioremediation potential. Finally, the objective of novel gene discovery. Metagenomics is key to finding the dark matter, or genes not in reference databases. This is crucial for identifying new enzymes and pathways for industrial applications. The methodological workflow involves 4 main steps: sampling, sequencing, assembly, and annotation, moving from environmental collection to functional prediction. One key impact of metagenomics is enabling the study of 99% of microbes previously considered unculturable, revolutionizing microbiology and biotechnology.